either this thing or these things.
I think of that. It's
Right?
gonna be right now.
That
It's
looks
that.
like it.
Yeah.
So talking to this weird square.
Oh, yeah.
So you can move to you can move that closer to you so we can capture your voice better.
Okay.
So if you use the desktop, here. Same.
Just feel for you.
I don't know why. I feel kind of dirty.
It's just like the time. You wake up like just 10 minutes later than you used to.
Yeah.
Yeah, I would say at this time probably up north in Canada is the best time of the year.
Right.
It's nice. Yeah.
Should we all go to
June.
Right?
Yes.
Should we go to Jasper and
My experience.
The air smells so good.
My experience as a PhD student is probably gonna make you all jealous because we all have to go to up north Michigan like almost every week. Drive there to sample, do the sampling.
Wow.
It's a long drive. It's like 8-hour drive for one single trip, and just come back the same day.
Really? You come back the same day?
Yeah.
It's only like 25 hours to Canada from here. It's just straight
Only
up.
like 20.
It's farther than that because it takes me 25 hours to get
Wait, what are you doing?
Forget
Hey,
ourselves
Neil.
in Colorado.
And then
You can use that.
where Paxton
We'll give you
lives,
that one.
It's literally
Yeah.
25 hours straight up.
Straight up.
And then to get into South Dakota or North Dakota, you're another 6 hours, and then you're probably another 5 hours into Canada.
So it's 36.
We'd have to
We're only 36 hours.
Only
We
if
can
we
make
take
it.
turns.
We're
right?
closer to
Drive.
it.
We're
Yeah.
closer
We
to
can do
Canada than we are to Salt Lake City.
Really?
Huh?
Really? That's how far
Wow.
the United States is compared to lengthwise.
Dang. What?
Yeah, it takes me
That blows
over
my mind
24
right now.
hours longer to get to Salt Lake than it does for us to get to South Dakota.
That blows my mind right now.
Yeah, because when you look at a map, it doesn't seem like that, but I've unfortunately driven it. It's
How long does it
long.
take from here to Salt Lake?
Salt Lake City.
Salt Lake City?
Yeah.
I think it's 36.
Wow.
Oh boy.
My mind is so blown right now.
Or do you go under
It have to. I guess it's gonna take us
don't
like
go over the Rockies.
more than
like
10
you go
hours to drive
under-
out of
Oh, actually,
Texas
no,
if
you
we
go.
drive
So
that
you
way.
go,
This is
I mean,
so
you go
huge.
this way, or you go
We're gonna start.
into Albuquerque.
Yeah. Oh, there we go.
And through,
So quiet down.
Yeah.
Quiet
Okay.
yeah, we stay.
Alright, you guys. So, well, I'm glad that we finally got this to work.
Wait.
Are they here? The other ones? I don't see the bar.
Yeah.
Can they speak if they can hear us?
Okay, do you guys see anything?
We can hear you.
Can
You
you?
have to you have to share the screen. That's the
Oh,
problem. Yeah.
okay, well. let's do that.
Fun. We're all the way. Share on the bottom.
Yeah.
Oh, there, yeah.
Share. Contact. Then we can just share your
Yeah,
PowerPoint.
Just a
No,
screen.
PowerPoint.
The one
PowerPoint.
below.
I
Yeah,
try to
yeah.
do this because, like,
Yeah.
because when I change it to the this presenter thing,
This one.
Mhm.
they won't be able to see it if it's just an application.
Oh,
Yeah.
really?
Yeah, so you have to just share the screen.
Ah,
I know because
there
I
you
did
go.
this before, and nobody
Really?
could see anything during my retreat thing. That was so embarrassing.
Aw.
Okay, now you guys can see it. Oh, but that's gonna block it.
Yeah, you have to keep moving that box
Can
around.
press that little arrow.
We don't want to see
Oh,
them.
okay, this one.
They go away.
There you
There
go.
you go.
Okay.
But you still have to put that little bar away from your title.
What do
Yeah. There you go. There we go. Ooh,
well, let's put it there over here, right?
that's fine.
Okay, now we're gonna start. Okay, so this is my first presentation since returning from the COVID times.
Ooh.
Wow.
Wow.
Yeah, I know, it's a pretty big deal. So I wanted to bring you guys this interesting paper that I found because it kind of reminds me of a lot of the stuff that we do in this lab. First of all, there's human tissues involved and there's zebrafish, okay, except we're not doing microplastics, we're doing PAHs, okay, and not just any old PAHs, the ones that have been degraded by the sunlight, the PAOHs, okay? So this is the study in vitro and in vivo Genotoxicity, so DNA damage is what we're talking about, of oxygenated PAHs by McCarrick from 2019. Okay, so before I start, just in case we don't know yet, oh, come on. There we go. Okay, so these are PAHs, polycyclic aromatic hydrocarbons. You see, like, aromatic hydrocarbons are just these ones with the rings. Okay, in contrast to aliphatic hydrocarbons, which are just the chains.
Is this really from our Hussein here, the paper?
I don't know. It could be.
Hussein Abdallah?
No, I don't think so. No, I just found this paper online. And it had a nice picture on it. I don't think it's from...
So he can't make that nice picture, I'm pretty sure.
Yeah, probably. He doesn't even do PAHs.
Oh, I thought he did that kind of stuff.
I don't even know.
Yeah, okay. Well, anyways, PAHs, right? Okay, and then, like, through a variety of different forces, such as, internal metabolism could do this, but, the one that we're looking at is, like, sunlight. Okay, photodegradation causes them to react, and, they might add on some other elements, like sulfur, oxygen, or nitrogen, and become other compounds like these. These are quinones. Okay, they all have these oxygen groups, these are carbonyl groups, right? And so these carbonyl groups make them more hydrophilic, possibly more bioavailable. So if that's the case, then they could be more toxic. All right. So this paper is going to study both of these, right? And they're going to be getting them from soil. Okay. So soil is very commonly used to study for these toxins because they're little fine particles, the low surface area. They adhere a lot of these organic contaminants to them. Okay, so we need to get the organic contaminants off of the soil. So the soil that they're interested in was taken from this wood preservation site. I don't really know what that is, but if there's a factory there, there's a high chance that there is some sort of pH contamination because factories use fossil fuels and incomplete combustion fossil fuels cause pH to go in the environment. Okay, so to get PAHs and other organic contaminants from these soils, they use a process called PLE, which is pressurized liquid extraction. It's also known as ASE, which is accelerated solvent extraction, the same thing, right? The way they do it is they take a solvent, which is DMSO in this case, and then the DMSO is going to try to dissolve the organic contaminants from the soil, okay? But they're stuck to the soil, so it's hard to take them off the soil. So to do that, they increase the heat, right? If you increase the heat, you can increase the extraction rate, but then if you do that, they might change state because these PAHs are so volatile. So to prevent them from changing state, they add pressure, okay? So they pressurize it to force it to stay inside the solution while adding heat, okay? So that way they can take it out, and it's apparently it's a very popular method of getting the organic contaminants off. Okay, one thing that I'm really curious about is, I wonder why it doesn't chemically react when you're adding all that heat.
Chemically react with what?
Just like they might, or like oxygen, I guess, or maybe that's why they use nitrogen here to make it inert.
Mm-hmm.
Yeah, okay, that makes sense. Keep touching it. Okay.
Wait, what does that have to do with heat? I'm confused.
Oh, sorry. Like they only react with heat if there's other compounds available, such as oxygen, which I guess in this case it's sealed off, so that's probably why.
That's why they use the inert gas.
Yeah. Okay, so they get the things off, and then we get a bunch of these hydrocarbons, right, or some other organic contaminants. So once we collect our hydrocarbons here, then they separate into two different parts. Okay, they do fractionation, soluble and insoluble part. So that's pretty easy to separate. This one, they're just the PAHs, and then this one is the soluble, the PaOHs, the quinones, the oxygenated PAHs. Okay, so then they're going to test both of those separately and together, combined in a mixture on.
Is that hexane acetone
Which one?
extraction, this insoluble soluble, to separate these soluble and insoluble? Oh, it's typically hexane and acetone.
Okay, it's typically hexane acetone extraction.
I was just
fractionation.
Okay.
Yeah. Okay, so they tested on these. Now, real quick, I just want to ask you guys: does anybody recognize any of these three things?
Yes.
Yes, of course.
Yeah.
What are they?
Beautiful goblet cells.
Yeah.
It's touchscreen, huh?
Okay, so what kind of tissue is this?
Epithelial.
It's epithelial tissue, right? So it's got those
We
different
learned
layers.
from Wei.
Yeah, and then what animal is that?
Just
last one?
realize how boring this is.
Chicken.
I'm seeing it down here.
Yeah, this is freaking boring.
Boring.
There you go.
Yeah, and
Hey,
then
Kayla and Chi, you guys have to register for this class for next semester, okay? Just keep that in mind. For sure. I'm
Yeah.
serious.
okay, so yeah, this is epithelial tissue. It's from the bronchi. Okay, and the reason why they chose bronchi is, they suspect that inhalation is a big source of human exposure to these chemicals. Okay, the second one is the liver cells. Okay, these liver cells with the cancer cells on them, the hepatocarcinoma, and hepatocarcinoma cells, they have this protein that they want to look for. Okay, the protein that they want to look for is called CHK1. I'll explain that a little bit later. Okay, and then the last one is zebrafish embryo. Okay, so they did some tests on that one as well. Okay, so what did they look for in all these three things? Well, they primarily look for DNA damage and also abnormalities with DNA replication. Okay, so DNA damage and DNA replication. And the first one is micronuclei formation. So like if you have some DNA and they're trying to replicate, and some of the pieces of the DNA they're like messed up, so they don't get replicated, and they collect themselves inside their own little nucleus here, micronucleus, and then so that's a sign that there's a problem with the DNA replication process. So if you detect micronuclei with flow cytometry, then there's something wrong with the DNA and also the replication process. Okay, the second one is just a Western blot technique to look for proteins, and in this case we're looking for CHK1. CHK1 is called checkpoint, and it's basically a protein that looks for problems after post DNA replication. Okay, and it tries to fix problems as well. Okay, and then so the last one, common assay, is a very common and basic technique to look for DNA damage. Okay, so as I explained last time, common assay is basically you're taking the DNA and you're running it through electrophoresis, and then it causes the DNA to travel, right? But only if it has short enough fragments to travel, okay? If the fragments are too long, they don't travel through the gel. So, see these ones with the tail? They travel, right? So it starts like this, and then eventually they travel, okay? And the ones that travel the furthest means that it's the shortest fragments, and the reason why it has so many fragments is the strands broke, okay? The strands break means DNA damage. Okay, so increased DNA damage means it's going to exhibit more of a comet tail. Alright, so these are the things that we're looking for inside those three different organism things, like the cells and the zebrafish. And so let's see what they find, right? So the first thing that they find is inside this table, right? If we look, the first thing I want to show you guys is how this table is organized. It goes by dose, right? So 0.1 to 1 to 10, it says somewhere, but I mean, I'll just tell you, it's micromolarity. So let's figure that out real quick. Micromolarity, right? So let's just talk about regular molarity. Regular molarity is moles per liter, okay? And so 1 mole per liter. Okay, 1 mole per liter is pretty high. How much is that? Well, most of these PAHs are they're like 200 grams per mole. So that's like 200 grams in 1 liter. Okay, so remember this is micromoles, so that's actually 200 milligrams per liter. Okay, so 200 milligrams per liter is 200 ppm. That's a lot higher than what the numbers that we're using, right? So 200 ppm, 2000 ppm, 200, sorry, that's not right. 200 ppm, 2000 ppm, which is 2 ppt. Okay, and then this one, the lowest one is 20 ppm. Okay, so the doses are pretty high in this case.
What do you guys use? Not to interrupt.
It's like 0.1.
Yeah.
We use
ppm?
ppm,
Yeah.
right?
Yeah.
But
Wow.
They use molar.
Well, I mean, it doesn't really matter which unit they use, but if we translate it to convert to the other one, this is like 200 ppm.
Right, but it also depends on the molecular weight.
Yeah,
Right.
but I'm just saying, like, most of a lot of the PAHs are 200 grams
But
per
there's
mole.
probably a huge range, right? Did we look at the range? Or we
No.
don't care for the point of this?
I mean, I don't care either, so.
Cool. So Sorry,
Yeah,
didn't mean to interrupt. I was
It's
just
okay. I
wondering.
just know that like a lot of the pHs are roughly around like 150, 200 grams per mole. Right. Okay, so that's the dose we're using. It's a lot higher than what we're using, and just try to look for trends across because it's increasing in dosage in a log scale, right? So what do we see, right? If we look at the first half, there's no like. There's not much of a trend because sometimes the size of those flip, but at least we can notice that most of the lowest concentration has higher viability than most of the highest concentration. Okay, it's not always the case, like this one is lower in the first one, right? But most of them, right, are higher viability when in the lower doses. Viability in this case is defined by the cellular respiration process. Okay, so if they're doing cellular respiration and metabolism, then viable. Okay.
We're trying to. How can it get that high concentration in cell culture?
I don't know.
What's the Alamar blue? Like I'm not familiar.
Alamar blue is a test to look for
Viability.
presence of metabolism.
So is it different than MTT assays, or is it like a similar?
I don't know an NTT.
It's slight. Well, here's the thing: the people always kind of confused by viability and proliferation. So actually, they're two different things.
Yeah.
But whenever people talk about how to measure the viability, they always think about either use MTT or the blue, the Trypan blue, what they use.
Polymer.
The Alamar blue. I think that's like just like Trypan blue. Only the live cells take the dye or something.
So it's like a
Also,
colorimetric
the live death
absorbance.
stains, not metabolic.
Well, they use that as a indicator for the viability. And it technically this is nothing to do with proliferation, because proliferation is the potential for cells to split,
Yeah.
not just alive.
Yeah.
But when people talk about this, a lot of people just think they're the same thing, so they don't really have a very clear definition of that kind of stuff. So
But that's like, this is similar to MTT.
I just don't understand how
What's
can they
the-
get such a high concentration in the cell culture. Unless they don't use the cell culture as a medium.
Well, the concentration in DMSO is pretty high, 0.1%.
Right, but do they dilute them with the cell culture medium? They should, right?
Maybe.
They have to, because they can't use the DMSO directly culture the cells. They're gonna kill all the cells.
I think, well, that's like the control. So in DMSO, the percent viability is still like 100.
No, this is not in DMSO. This is
Oh, no, the
DMSO
very-
added to the cell culture.
Yeah, like the very, But is that their control? They're saying like,
Yeah.
in case there's like trace DMSO in the
Yeah.
But
pHs
look for the control. Do they have like 1% or 10% of DMSO added to the cell culture? They don't have that.
But no, it's 0.1% DMSO for all of them.
0.1%. Oh, for all of them.
Yeah.
Oh.
But can they? I still don't understand. If they have such a high concentration in 0.01% of DMSO, if they added it to the culture medium, that's equals to dilute it with the cell culture medium.
Oh, so yeah, because it's like the pHs are already diluted in DMSO,
Yeah,
and then they
pHs are
dilute the
DMSO.
DMSO pH in the cell medium. Maybe.
Right,
Yeah.
but
Yeah.
once they get into the cell medium, they can't reach that high concentration. That's a problem.
No, they can't because these are quinones and
Those are all water soluble.
the quinones are highly water soluble.
Except for the benzoylpyrene, the first.
Oh yeah, except
Yeah,
for
that one.
Wonder how they did that one.
Right, it just.
You startled enough with it.
Does it say how they did it? Did they treat like we do?
You guys can read it. That wasn't something I was
Supposed to be
interested
telling
in.
us about it.
Well, I'm telling you about a bunch of other things that I found important.
Well, we'll check. We'll double check that after this. Go ahead.
It's not that important.
Well, it's important for us because we're doing this, so we needed to figure out how exactly they can reach that high concentration.
see.
I'll
Yeah.
pull it up if you want to keep continuing.
But that's
I'll
fine.
see if I
Just
can
go
find
ahead.
it.
Okay.
Also Zerman.
Alright, so anyways, the cell viability, right? The viability appears to decrease by increased concentration, okay? If we look at the other side right over here, so those are like survivorship, and we can't really see much difference. It's about the same. So let's just consider that one to not be significant.
So apoptotic and necrotic
Yeah.
cells. What the
Non.
what? Right. What assay they use?
They use flow cytometry and then they use that resurrent, what is that, resurrent blue?
Alamar blue.
Yeah, the Elmer blue.
It
That's
says for
resurrent blue.
2-color flow cytometry for percent non-apoptotic necrotic cells.
Okay.
They should look for, oh,
Two color flow cytometry.
yeah.
I don't really know how that one works either.
I guess you can separate by the cells that were stained or not stained.
Yeah. Here's another thing.
Mhm.
Apoptosis and the necrosis are totally different.
No,
I think.
you have to do an
This is-
assay
It
for
says
that.
non-apoptosis.
Right. It's not apoptosis and non-necrosis.
Yes, they combine them.
Right. So when you see the necrosis, the cells are probably not gonna even be seen in there. They're like dead. But when the cells go to apoptosis, their cell morphology might be still there. You can see them, but there are a lot of changes
So
happening
I guess they
inside.
just combine both
Yeah, you
because
have to
if
do
you
like a
just
task.
Right.
even if you discounted one of them, the numbers may have been very strange.
Okay.
Like not there.
no, as in like very variable or something,
How do they have 102 plus or minus 3%?
or 102.
That
Mhm.
see on the left?
Percentage
And then
of
how about this
viability.
thing?
106 plus or
Those
minus
ones,
3.4.
I mean, those ones those ones must have proliferated.
That was gross.
Those ones must have like started cell division. You know, they like the 0.1.
Little PAH.
Yeah.
It's actually good for you.
Little cookie.
Cancer though, I mean.
Sprinkling of it.
Microdosing.
I don't know, I'm sure they have an acceptable reason. Like, we shouldn't pay too much attention because they're not statistically significant. So
Yeah. Go ahead.
yeah, okay. So the next one is, let's look at down here. Okay, so down here it shows a much clearer trend between the 3 different doses, and we see that right here the pH fraction, which is the insoluble one, and then the soluble fraction, right, shows about the same. But the point is the clear downtrend. There's more of there's less viability at the higher concentrations, and then the mixture right here did not show much difference in all of them. So I guess the mixture is just really toxic, right?
Yep.
Even at the lower levels. Okay, cool. So what do we get from this? Well, we get that bronchial cells are less viable at higher doses, and there's not much information on the survivorship.
So I guess they don't really mention or care about anything like realistically what are the concentrations out there for in the environment, because they're using a very high
They
concentration.
probably only reported the concentrations where they could see
See the
something.
difference? Yeah.
I'm just guessing.
I think that's very
Probably
likely.
below that, they
Yeah.
don't see anything.
Yeah, that's kind of strange. I don't see someone breathing that much in.
No.
No, it's hard. Well, for 0.1 milligram per mil,
Yeah.
wow, you almost have to stuff the person with that, in the airway. It's yeah.
I'm just kidding. I guess the people who work at the wood processing plant, they could inhale it. Because that's
Very
what they do. They
extreme.
spray PAHs to make things water resistant.
Or if you were like a firefighter or
Yeah,
something.
yeah.
That's
It could
possible.
probably build up over time in your tissues. too,
Yeah.
With like chronic
Imagine you
exposure
do that
versus
every
acute.
day. Yeah,
Yes.
maybe.
But yeah, still, I would say this
Yeah.
kind of concentration is crazy. That's gonna kill a person.
Yeah.
Yeah. Crazy, man.
I hope they use
No, I mean, you'd be surprised it only killed 5% of the cells.
That's what I'm saying. Like, so the cells are really
It is
resistant
like-
to that.
Okay, the exposure time was 72 hours, by the way.
Wow.
Okay, so anyway, let's move on, right? So this next table or this next chart right here basically shows the bronchial cells again. Okay, and the thing that I really want to show you guys, if you look at the different colors, those are the doses. Okay, so there's a clear dose-dependent relationship
Wow.
for all of the almost all of the chemicals, right?
For the DNA.
Yeah, this is DNA damage, okay? So, common assay DNA damage. Alright, so the DNA damage increases by concentration of any chemical, pretty much, okay? And so,
But
Hmm? What?
there's only that one that's statistically significant. The other ones are within the error bars, overlap with the error bars. See
Yeah.
that bottom B chart and then the last one on the right. So
yeah, the last one, so the really high concentration one.
yeah.
Yeah, so the really high concentration one appears to be the only one that matters. So that means that the low concentrations were all good. No.
Okay.
We're fine.
No
Don't worry
regulation.
about
Put that at the
it.
end of your manuscript.
Yeah,
We're all
there's
good.
just there's just no significant difference, right? Okay, good observation there. Okay, so we notice that DNA strand breakage increases with your dose. Okay, for the bronchial cells, but we can notice some other things here. Like for example, if we look at this one right over here, we see that the mixture is worse than each individual pH or the oxy-PAH.
Yeah.
Okay, and then those two are about the same. Actually, there's no significant difference between them at all. Okay, and then what we can notice on the top is that out of all these quinones, the non-quinone benzoylpyrene actually shows to cause the most DNA damage compared to all those other quinones in the bronchial cells, which is very interesting. Okay, so we're going to see a little bit different results in the next one. Okay. Okay, so this next one is looking at the zebrafish now. Okay, so the zebrafish DNA damage, pretty similar results to the last one. Okay, so what we can notice from these diagrams is, well, first of all, look at how the x-axis is arranged, right? It's again the dosage of either the PAH or the PAOH, and then as long as it has a positive slope, then it's dose dependent, right? Increasing DNA damage with increasing dose. So you see all of them are going up, they're all increasing, they're all positive slope, so that means that it's consistent with the previous results. Okay. Well, there's something else that we can notice here, which is the fact that in all of these, right, let's look at the colors of these markers, right? The black square is DMSO, the control, okay? And then the black circle is the PAOH, the empty circle is the benzoylpyrene, okay? And this is the mixture, the half circle, right? The semicircle. Okay, so what do we see in all of them? Well, we see that the benzoylpyrene is causing the least amount of DNA damage this time. Okay, previously it was causing the most, right? It's causing the least amount of DNA damage in the zebrafish embryos. Okay, what's causing the next? Well, the next one that we can see is, that's the black circle, right? The black circle line is the PAOH. So the PAOH causes more damage than the benzoylpyrene, right, in zebrafish embryos. Okay, and then the highest one is the mixture. Okay, so in accordance with the previous result of bronchial cells, the the mixture caused the most DNA damage than the other ones. But the difference is the benzoylpyrene and its derived, oxygenated pH is flipped. Okay, so in this case, it's really interesting, right? Sometimes the pH is more toxic, sometimes it's the other way around, right? So maybe it depends on the species. Okay, so the next thing they looked at is DNA damage or problems with the replication process due to the micronuclear formation.
Wait, so benzoylpyrene could be a cancer treatment because it didn't break DNA in the system, in the whole system, in the organism, but it killed the cancer cells, maybe? No?
Not really.
The benzoylpyrene, no, I mean, it still causes more DNA damage than the control, you see. So,
Is the control
Where is
just
it?
DMSO?
They
yeah,
did,
the control is
DMSO.
did they plot the control all the way across or no? Just the
You
top?
just have one.
No, the control is
One
just.
Oh, they just put it there, okay.
I wonder if that has to do with the solubility again, like with DMSO and the benzoylpyrene versus the quinones.
Oh,
Maybe
it
that's
doesn't
why
matter
it shows
because
up so
they
similar
got
to
the-
it.
They still
They
did.
set them.
Did they use DMSO to dissolve them? Is that why
Yeah.
they use DMSO as the control?
Yeah.
So that's how they got the high concentrations?
Oh, that's what I think. But then,
Yeah,
it's
that's
the-
No.
This you have to dissolve this. So for example, if you want to treat the fish, you can't put the fish into DMSO, like any percentage of DMSO. That's it's not gonna happen. So they have to dilute them DMSO really like many folds.
Okay, well, okay, let's just figure this out real quick because everybody cares so much. So, look, the solubility in DMSO is, pretty reasonably high. Okay, it's like what, 1 part per thousand or something.
Right.
No. It's more than that, I think. Okay, it's pretty high. And then so if it's like 0.1%, that's only divided by 10,000. That's only a 10,000 dilution.
So 0.1% of DMSO, they use that to dissolve
Yeah.
the chemical.
Yeah.
But if they use 0.1% of DMSO to dissolve the chemical, can
No,
they still reach
no.
to
It's not 0.1% DMSO. It's only 0.1% in the final.
in the final
Yeah.
concentration?
So
But
like,
that's the issue. When you
use the
dilute
pure
them,
DMSO.
there are a lot of precipitation gonna come out. They're
Okay.
just not gonna stay in there.
It goes like,
They said
it goes like that, so you have your PAH in the middle, and then the DMSO is gonna go like this, and it's gonna surround it, and then these O's on the outside are gonna make it water soluble. Does that make sense? So there's a dimethyl sulf oxide.
Yeah.
Compared
this
to
is John
the
So
other
it doesn't matter how many times we dilute them.
This is how it makes it water soluble, cause it will go, does that make sense, everyone?
Yeah.
It'll go around here and then water will
Now
be
it has
able
all
to
these
follow it.
available oxygens.
Yeah, so then H2O can hydrogen bond here. So it just makes
Makes
this
sense.
chain around there.
No, that's not the problem. The problem is Dr. Xu doesn't think that the final concentration could be 200 parts per million, given the fact that you had to dilute it 10,000 times. Right?
Right. So my experience is if I use say DMSO or ethanol to dissolve like the chemicals with a very high dose, after I put them into a medium to treat the cells, we're gonna see the precipitation. They're not gonna stay inside the solution. A lot of them come out. So that's my experience. I don't know if Nin, do you see that like similar things happen?
Yeah, they precipitate.
Really?
So that's what I
Does it feel like a
Okay,
layer?
let's see.
No, they're just like the solid things coming out.
Oh, really?
Yeah, they're just not.
It becomes cloudy.
Does it
Very
start
cloudy.
doing that immediately, or does it take a few days? for
It
to start?
happens immediately.
Yeah.
Huh.
So that's why I don't know how they can reach that high concentration. Maybe.
Well, I mean, like they
Find a way.
dilute it by like 1,000 times. So it's 1,000, not 10,000.
I know.
That's up to 1 micromolar.
Well, how much DMSO did they use? That's part of it too, because the
0.01%.
more, if you have DMSO to surround every single molecule of your PAH, then every single molecule will maintain solubility in water or in aqueous solution.
They probably use pure DMSO to dissolve all.
Yeah,
Yeah,
DMSO
yeah.
solution.
Not the
Yeah.
DMSO solution, the
Yeah,
pure
I'm
one.
sure they
That's
did.
probably, I didn't even think of it.
Really, why is this so like mystifying?
But because I
It's
just wondering if
interesting.
we can have that higher concentration if we do that.
Yeah, we can, we just have to make these
I think we can,
the same.
because like 0.1% DMSO at the end is really high.
I know. Here, let me make this straight. For example, if you have a chemical with
Yeah.
1 ppb,
Yeah.
you use a pure DMSO with the 1 ppb.
Okay.
Okay.
Yeah.
After you dilute them 1,000 times, your target concentration is going to be 1 ppm,
Is it? Oh yeah.
right?
From 1,000 to
1,000 times.
million.
So that's what you're hoping. But you see a lot of precipitation, which means there's really no way that you can still have 1 ppm. Am I right?
If you have precipitation, yeah,
Right.
that's correct.
You gotta have a much lower concentration than that. So that's my
Only so
argument.
much capacity.
I don't know if they mention anything like that.
I see.
So that's my question. I hope the thing gonna work like this. We're doing the same thing. I really hope this is gonna work like this.
Well, I don't think there's a problem with most of the chemicals they use, because remember, like I said, the quinones are water soluble.
Yeah, quinones are fine.
Yeah,
I
but
used
the
quinones
benzoyl
before.
pyrene
But
one
the
is,
BAP, the benzoyl pyrene,
Oh, that one's a pain.
that one doesn't really have a very high concentration in my opinion. So we can give it a try. We have benzoyl pyrene, right?
Yeah.
We can calculate the structure in 3D, how much space that pH takes up in 3D space, and we can calculate how many of these DMSO we need to stick on the surface, and then we can get the concentration that we need to surround every molecule to make the solubility much higher.
Yeah, we can try
That's
it.
crazy.
We can try
That's
it.
so cool.
No, but we want to limit the amount of
Then you have like the
DMSO.
stock solution, and you can dissolve it in water once it has bound to the DMSO.
That's the
right?
only way you're gonna get the concentration that high,
Mm.
and be able to know that every molecule is surrounded by DMSO, because the DMSO is facilitating solubility through this mechanism only.
So Nin, this may sound like a tiny problem at this moment, but if you talk to Umbras, and they are also kind of struggling to measure or to have those ones dissolve in a very neutral solution for measuring concentration. They've been spending a very long time, a lot of times working on this. So it could be an issue if we can't figure that out. That's the reason.
Well, I'm using like low concentrations now, 0.1.
But how do you know that it's not precipitating out and you just can't see it because the concentration's so low? I mean, with high concentrations, it's easy to see a precipitate because there's more of it. But what if it's just too little for your eyes to detect?
Yeah, just think about the example.
Well, although it could be precipitating, I'm still operating within the bounds of its solubility.
Well,
on paper.
But you don't really know the exact concentration after the dilution. That's my point.
Yeah.
So that
Why are you so worried about this when you're not worried about the PAH is evaporating in the chamber?
Evaporating in the chamber.
Yeah.
Well, if you cover it tight, I think it's should be a.
Well, then fine. If I shake it hard enough, it'll
No, this
also
is something.
dissolve.
No. You're shaking. This is something you can't really change once you get it precipitate. We have to figure out the actual concentration. That's for sure.
We can
Even if you don't know, later on we can use like the
The
GCMS or the LCMS to figure it out. But we have to know, otherwise we can't write in the paper. Like what are we going to say?
Be results if you don't have your concentrations. Right.
We don't know the concentration. We can't really write the results. So that's a problem.
So we have like the mixed constant, like we write down what we created or what we think we've created, but then save enough of the sample to do GCMS.
Yes,
Right. Okay.
Yeah.
definitely.
So we have to measure that.
Otherwise, everything's
Yeah.
worthless.
Which is why it's important.
Yeah.
Which
But
makes
go ahead.
total sense.
Alright. Fine. Let's move on. Okay. So, are we okay with this? Pretty easy to understand. Right? Zebrafish embryos seem to be affected, their DNA is affected by increased concentration, but this time the benzoate pyrene does less DNA damage than the associated quinone. Okay?
Sorry, just one quick question. How long were these exposed for? Do you know?
72 hours.
Okay, cool. Sorry.
Yeah. Okay, so this next one is looking at the problems with the DNA replication and looking at the micronuclei, and we don't really see anything. Okay, so in this case, that means that the different doses of the PAHs and PAHs or PAOHs themselves compared to control, even they don't really show any difference. Okay, so there's not much significant difference. There's like a few, right? But for the most part, there's no significant difference in micronuclei formation, so it doesn't affect the DNA replication as much as it does damage it. Okay, the last one, it doesn't count. That one is a positive control. Okay, so that one's supposed to be high. All right, so that's what we get, and let's move on to the next graph. This is the last one. This one is the Western blot looking at the presence of the checkpoint protein. Okay, and then we see very similar results to the bronchial cells. So this is the liver cells. Okay, the liver cells in the human show similar results to the bronchial cells. First of all, we notice that there is presence of the phosphorylated checkpoint. Phosphorylated means that it's being expressed, and there's gene expression for that in all of these treatments, but most in the benzoate pyrene. Okay, so that's a very similar results to the DNA damage one.
So I guess CDK2 is
CDK2 is a positive
housekeeping.
control.
Housekeeping.
Yeah, or it's just the this is a protein that is associated with having cancer, and it's gonna cause it's gonna be increased if those happen. So that means all of these things cause cancer.
So I guess this is the CDK2 is important to generate the the figure on the right, because on the figure you have to normalize your data for the CDK1. So and that's how you actually do that.
Okay, that's good to know. Thank you.
Why is the control so high on them?
This kind of
No, the control's like a little lower than that one. See how dark this is compared to that one? So it's a little better.
I don't think it's convincing.
which one are you talking about?
Both of them, A and B, the
we're
control
talking
well
about
as
The
Looks like-
fact that this one is thinner than the other.
Yeah, I guess it's a little bit thinner.
No,
Yeah,
no,
it's-
the CDK1 is supposed to be similar all across all the samples because that's control housekeeping.
Housekeeping.
So we use that housekeeping to normalize the results.
Oh, okay, okay.
Right. So this is like the gap pH or
Yeah.
actin. Yeah.
Wait, so this has nothing to do with those ones?
It's nothing to do. It's just to help you to normalize the data.
Oh, I thought
So-
this was the.
Yeah, so everything's that minus.
I thought this was the existence of CDK2 with all this.
So that's why you see the bar graph. You only have the CDK1, so or CHK1.
Yeah.
So the CDK2 is internal reference to help you to normalize the data, so that they don't make any of the bar graph for that.
Yeah, but the reason why it showed up was that this cancer-associated protein is present in all 5 of the cases.
Well,
Yeah.
if that's the case, it's you know it's nothing to show about. But they use this as an internal control.
Yeah.
So when they measure, so you know how to generate that bar graph. So when they use the image,
Yeah.
they measure the intensity
Yeah.
of the signal, but you have to use the ratio of the two different proteins to generate that. So that's how they do it.
Okay, so anyways, at least we can see that benzoylpyrene has significant,
Okay.
the checkpoint expression compared to control and the others, the quinones. Okay, so even though the quinones appear to be a little higher than the control, it's not that much higher. Okay, all right. So what do we get out of all this? Well, we get the fact that the DNA damage occurs to increased dosage in both the human and the zebrafish cells. Okay.
They never saw micronuclei in anything?
Yeah, the micronuclei was like increased in a few of the quinones at only like one of the concentrations. Okay, the toxicity of the pHs and the pOHs is dose dependent, and then the last one is the toxicity could be species dependent too.
What's the time that it takes for these cells to split? What is that called, time?
The turnover time.
Like the
The
Doubling time?
epithelial or the liver
Doubling
cell?
time. Cause if it was 72 hours.
no, it's not that long. I
They
think
use
regularly,
treatment
if if you grow them cells without any treatment, they should split like overnight, at least the
Okay.
one pass overnight.
And they,
They used within 20 passages.
20? That's
Well,
pretty high.
that's like when they started using the cells, all the
Okay.
cells are
The whole
within
time.
20 passages, so it's
Okay.
not very old outside. So, but still, like 20 is kind of beyond my standard.
Yeah.
I would like to use anything within 10 passages.
All right, guys. Well, thanks for this.
That's the end?
Yeah.
You left us hanging.
Huh? What'd you say?
There's a lot of debating for this.
Okay.
Well,
Yeah,
that
maybe
was
it
good,
was
though. It's very
distracting.
relevant.
It's good.
Thank you for
It was
presenting
a
that one.
nice
Yeah.
presentation. I liked
Why
all your
did it
steps.
Suddenly just end.
Thank you.
I feel like there should have been more.
Well,
Timing.
feel free to read the thing.
Any questions, comments?
You guys have a lot of questions. I think I'm
We
done.
do, because this is really
This
related
was
to
cool.
what we're doing.
Yeah,
So
was
that's
really good.
that's why we have a lot of questions for
Your
this,
slideshow
especially
was
for
very
the
good.
treatment.
Mhm.
Because believe it or not, there are already a whole lot of study for PAHs and PAOHs and
Yeah.
how they're gonna possibly be the pathogenic contaminants in the air. There are a lot of studies going on there. So that's why when I look at this, the results you show, there's some of them might not be consistent to whatever has been reported before. Especially when you show that there's really no micro nuclei in there.
Well, I mean that was just the bronchial cells.
It's bronchial cells.
That's it.
That's possible.
Okay, they didn't like do it on the liver or the zebrafish.
So they use I believe they use a cell lines.
Yeah, cell lines.
What's the name of it? What's the name of the bronchial cells?
Bronchial epithelial. That's it.
HBEC.
Oh yeah, human bronchial epithelial cell, HBEC.
So did they get them from some vendors,
Probably.
ATCC?
Yeah, I think so.
American Type Culture Collection.
ATCC.
Rockville.
Okay.
Yeah.
So this is a typical cell line.
Dang, gosh, she knows everything.
No, I'm reading the paper.
No, but he knows all those companies and stuff.
Yeah.
No,
That's where you've always
they're
been.
both from.
Because here's what I heard from our collaborators. So I happen to know someone working on the Johns Hopkins Hospital. and she's really like expert for respiration. And she kept telling me the cell lines we're using are very different than the primary culture they have from the patients. So first of all, when you look at the epithelial, like the bronchial structure inside, you're supposed to see a very well stratified cells, right? So what type of the epithelium on the inside?
Stratified epithelium?
Pseudo stratified, right?
Yeah.
So if we use the cell line, we cannot stratify them at all. So that's what they said. I don't know. I don't know if with this much of the difference, how do we kind of address the relation of our findings and the actual happening?
Right. It's not
So
very related.
I hate that we can't use the cell line has such a big difference compared to the primary culture, but that's actually make their lab had a whole lot of NIH grants.
Really?
Well, because imagine how many labs can actually access the human epithelial
Like hardly
tissue
any.
from their bronchi.
From
It's hard.
actual
It's
bronchi.
really hard. So
Someone in
Jen and
the
I
lab
were
is
trying to figure out a
very-
way if we can actually use the cell line to repeat this whole thing, but we haven't started yet. We're just kind of debating. Yeah.
Could you use several cell lines to create like something that would be representative of an actual primary tissue, like mixing?
Skin cells
Right.
are okay. You can stratify them.
Right, but
Yeah,
like
but
bronchial
for
ones?
bronchials are hard based on them. They said there's no way that we can stratify them. We never tried ourselves.
Try it. Maybe you guys
Right.
will figure out the way to do it, and then you all
You're
become
gonna miss the
famous.
For only for the primary cells, you have the ciliate on
Yeah.
on the top, not
You
these
can't
guys.
do-
Yeah,
We
I
can
hate
use
that.
like Shang's flow through to like stimulate movement.
That's the plan.
Yeah.
That's the
Thing,
plan.
Eating.
He's trying to build a kind of in vitro device to mimic that,
Yeah.
like the airway of human.